Hi, welcome to the Bioinformatics Chat. Today my guest is Luke Anderson-Trocmé, who's a PhD student at McGill University, and we will be talking about some issues that you can run into when using some old sequencing data. Luke, welcome to the podcast.
Hi, thank you for having me.
So we'll be talking about this legacy sequencing data, as you call it, and specifically the data that comes from the 1000 Genomes Project. So maybe let's start with some general background. What is the 1000 Genomes Project?
Yeah, so the 1000 Genomes Project is a collection of genome sequence data from 26 populations. This was a large kind of collective effort done by a whole bunch of institutes from all around the world, like 12 years ago or so. And this was kind of done over the course of several years using different sequencing technologies. And this was done, because 12 years ago, sequencing technologies were still being developed. So as they were producing the data, they were kind of developing things as they were going. I mean, this is one of a few publicly available data sets. And as you can imagine, publicly available data is a lot easier to work with than some of these private databases. And these databases are often used for all types of research projects. Anytime that you're doing some medical or population genetic research, you want to know how common a certain mutation is in a population, or you want to know how related two populations are. And so the 1000 Genomes Project is an excellent resource for, I guess, kind of figuring out how diverse human populations are, and just kind of as a reference cohort to kind of figure out what does human diversity look like. And many people will be using these reference cohorts for doing allele frequency estimations and a few different things.
Yeah, so as you say, this data can be used for many different purposes. And what was your purpose when you just started this research? You didn't start it with the intent to find some problems with the data set, but you started with some, normal research goal, right? What was that?
Yeah, so we actually started this project trying to replicate a really interesting result that was identified by Kelly Harris and Jonathan Pritchard, where they noticed that when you're looking at different populations in the 1000 Genomes Project, you can identify different rates of certain mutational profiles, and they found that the Japanese population of this database had a heterogeneous signature. So that means that like part of the population had a mutational signature and the other part of the population did not.
Now let's define this right away. So what is the mutational profile or a mutational signature?
Yeah, so basically if you count all of the mutations in somebody's genome, you can then categorize them into different groups. And so for example, if you take the base before and after every single mutation, you can classify them into what we call, three mers.
So we're talking specifically about point mutations here.
Exactly. Yeah, we're talking about single nucleotide polymorphisms or SNPs. And I guess an example that I like to use, because people tend to be a little bit more aware of this kind of thing, is these mutational profiles can be used to diagnose certain types of cancers. So for example, like UV radiation will cause thymine dimers in DNA, so that it will only affect two T bases that are side by side, and it's going to force them to bind together instead of across. And what you end up getting is that if you have a cancer caused by UV radiation, like in melanoma skin cancer, you would have an enrichment of a certain type of mutational profile. In this case, it would be the TT thymine dimers. These are mutational signatures that are relatively easy to identify because cancers have a lot of mutations. But the ones that we'll be talking about are more subtle mutational profiles that can only be identified at a population-wide scale. So we're looking at tallying up all of the mutations for an individual, but then also doing that for all the individuals in a population, and then comparing, what are the enrichments of certain types of mutations across different populations.
And the idea would be that, as in cancer, these signatures have a specific signature might correspond to a specific source or a specific mutagen.
Yeah, exactly. And so this is still something that's being discussed and kind of looked into as to what exactly might be causing these differences in mutational profiles. There would be, some have suggested a possible mutator phenotype, or a slight mutator phenotype, meaning that like your DNA repair mechanism or something involved in that might have a slight bias towards not being able to repair certain types of mutations. Now, obviously, this would be, fairly limited in how many mutations can occur, because of course there's going to be negative selection against any mutator phenotype. But the biological reasons for these are not yet fully understood. But you would also presumably have many environmental factors that could produce something like this. So, for example, if the population in question has elevated smoking rates or elevated exposures to certain mutagens, you might expect to find possibly an increase in a certain type of mutational signature in that population.
Right. So you were talking about this study by Kelly and Pritchard, and they discovered this specific mutational signature in the Japanese population. Now, what was their discovery based on the 1000 Genomes Project?
Yeah, so in 2017, they identified a mutational signature that was enriched in only a portion of the Japanese population of the 1000 Genomes Project. And so I should say that Harris has also identified multiple other mutational profiles in other populations, and those mutational signatures have been replicated in other databases. But this particular one with the Japanese population had not yet been replicated in another population, in another data set than the 1000 Genomes Project. And so it was an interesting mutational signature because only part of the population had it. And the reason why this is interesting is that because we're looking at genome-wide mutation rates, if there is population structure, so that would mean like limited mixing between individuals in a population, it would take a long time, many generations, for a genome-wide signature to actually be differentiated between these groups of people. So it seemed a little bit unlikely that it was a population structure effect, especially in the population of Japan, which is on an island, a relatively small island with a relatively high population density. It seems unlikely that there would be such strong population structure. The other hypothesis that we had of how to explain this effect was maybe there's some environmental factor here that might be causing something, and Japan has had a history of mutagens that would have been exposed to the population, and so we were interested in trying to figure out, well, is it a population structure effect? Is it some environmental mutagenic source that's causing this mutational signature? Or the third option would be, well, maybe it's just a technical artifact. Maybe it's not a true signature that was observed in this population.
And why is the fact that the signature is present only in half a population, why is that weird? So if that was a single SNP, if that was a single variant, and we would observe it in a half of the population, we would just say that the allele frequency is 0.5. But here, is it because that when it is present, the signature can be present in very high numbers in a single individual, but that completely absent in other individuals? Or why is it that we find this weird?
Yes, that's a great question. Basically, it's that we're looking at a genome-wide signature, and the rate of these mutations across the genome is or at least in this database is relatively consistent. And so for a genome-wide signature to be preserved in a population, you would need very, very strict like lack of migration between these groups. Because anytime you would have any migration between these two groups, that genome-wide signature breaks down within like a generation or two, because you would have one parent from one subpopulation and another parent from the other one, and their offspring would have half and half, and that mutational signature would just disappear within a generation or two.
So again, is it because it's present in high numbers in a single genome, or what's the crucial difference between a mutational signature and a single variant?
Right. So this is it's a summary statistic. So whereas you're right in saying that it's not that unusual for a single mutation to be present at 50% frequency, that happens all the time. But what we're looking at is a summary statistic. So we're taking all of the mutations of a genome, all of the mutations of all the genomes actually, and counting the number of AAA to ACA mutations. And so there might be 50 of them. And then we look at the next category, and the AAA to AGA mutation might have 100. And so we're kind of summing up all of these different mutations to get a mutational profile. And so it's this profile that we're talking about, not necessarily one particular mutation or a select number of mutations. We're looking at the entire range of this spectrum. And in particular, the one that was identified by Harris and Pritchard, it's the blank AC to blank CC mutational profile that was enriched in the Japanese.
And that means that an A followed by C is likely to mutate to C followed by C?
Exactly.
And so you were observing that signature in high numbers in half of the Japanese population, but the other half didn't have that signature at all, right?
Exactly.
And so one of your hypotheses was that maybe that's an artifact.
Yeah, exactly. Or at least that was the one of the hypotheses that we had. And so it was either going to be, maybe this is if this is population structure, then wow, this is like a very strong signature and very interesting and worth looking into from a population genetics perspective. And so I work in a population genetics lab, so this was something that we wanted to follow up on. The other hypothesis would be maybe there's something environmental that's causing this mutational profile in the Japanese. And then the third option would be, like you just said, the technical artifact. And so that was the initial goal of this project, was can we replicate this finding that Kelly Harris and Pritchard found in a larger, higher quality Japanese data set. And so with some collaborators from Japan, we were able to get our hands on several thousand high quality, very deep sequencing from a few thousand individuals from Japan. And we found that none of those mutations were present in this newer database.
Mm-hmm.
And so that in itself was just, evidence suggesting, something funky's going on here. Something's not adding up. Maybe we should investigate a little bit further.
Right. So how did you go about that?
So the first step that we did, which is essentially what I was just saying, is that when we compare the frequency of mutations between the higher quality database and the lower quality data, or the 1,000 genomes data, I should say, we find that there's a large number of missing variants between these two databases, meaning that a mutation is present at a certain frequency in one database, but then completely absent in the other, which is not that unusual per se when you're doing these sequencing efforts, because there is going to be sampling noise where you're not going to be actually collecting the entire, population of Japan. You're only going to be able to sample a small number of individuals. But when we filtered out these positions, so we're removing positions that are missing in either database, so we're only keeping positions that are found in both. this mutational signature disappeared, and in the Thousand Genomes Project. And so that means that the mutations that are causing this signature are missing from the newer, higher quality database. And this was, we kind of thought, all right, we figured it out. This is a technical artifact in the Japanese population. Great, we did it. But then, we were kind of thinking, well, hang on a second. If this is something that's affecting one population of the Thousand Genomes Project, maybe it's also affecting other populations in the Thousand Genomes Project. And that kind of led us down this rabbit hole of trying to figure out the right statistic to use to be able to identify these mutations in these other populations.
Right. So one challenge that you encountered was that not every population has this alternative data set. So you got lucky with the Japanese because they had this larger sequencing project that you can get hold of, but that's not the case for all the populations in the Thousand Genomes Project, right? So you had to be sort of smarter about how you use the data.
Exactly. So with the Japanese population, we were lucky enough to be able to compare it to kind of a golden standard of here is a database that has 10 times as many samples, much better, higher quality sequencing data. So we had a reason to trust this newer database. But like you said, it's, not all of the Thousand Genomes populations have been resequenced or sequenced to from another sequencing effort. And so for that reason, we had to think about a way to identify these particular mutations that we thought or we understood to be potentially technical artifacts. And so we started scouring through all of the supplementary materials from the Thousand Genomes Project, reading all of these extra supplementary methods and, digging through all these files and folders of the Thousand Genomes Project website. And then finally we came across, and every time that we found a different metric or a different thing, we would do a regression to see, does this correlate with the presence of this mutational signature? And we found that the average quality of the mapped bases of an individual was very highly correlated with the presence of this mutational profile.
And when you say this mutational profile, you're still referring to the Japanese?
Yes, exactly. The blank AC to blank CC mutational profile.
So you were trying to find an explanation for it and then maybe generalize this explanation to the larger data set.
Exactly. And so we found that this average quality of mapped bases worked out really well and was actually a pretty good predictor for whether or not an individual would have these particular mutations. And this was not only just a great, moment of, wow, we found something that might explain it, but it was also, pretty strong evidence that there was something, suspicious about these particular mutations where, why would all of these mutations be associated or correlated with lower quality data? And that was kind of the moment that we started to realize, all right, it looks like there's something batch effect, some technical artifact that might be causing these particular mutations.
Yeah, I imagine there are very few legitimate explanations for that correlation.
Yeah.
And when you discovered it, maybe you thought, well, how come we didn't think about this earlier, right? Because this is sort of maybe obvious in retrospect, in the hindsight, but I imagine, yeah, it took some time to arrive at.
Yeah, and I mean, I should say that, the quality control done by the Thousand Genomes is very solid. They have experts that have, done a very good job of actually flagging a lot of these regions that are not very, that they're not called confidently. And so there's a strict mask that exists. And so a mask is referring to regions of the genome that meet a certain criteria. So you might have like an indel masks where you'll just remove all of the insertions and deletions of the genome, or you'll have what I'm talking about is the strict mask, which is the regions that are kind of deemed high enough quality to be included in genomics research. And so the Thousand Genomes Project has been updating this strict mask over the years and kind of calling, these regions are trustworthy and these regions are not trustworthy. So they have been doing quite a bit of quality control over the years. But I guess, yeah, just doing a regression on the quality of the data to try and identify variants that are associated to quality was something that hadn't been done yet.
Yeah, that's a very good point. That's easy to miss. So the bases that we're talking about, they're not complete junk, right? They meet some minimal quality threshold. And do you remember, by the way, what sort of, like, what's the minimum, let's say, Phred score threshold the Thousand Genomes Project adopted? And when you run your regression, what's the threshold, more or less, of the bases you can trust versus the bases that can cause this kind of artifacts?
Yeah, so we actually, in the association test, we didn't actually have a strict cutoff for quality. But if you look at the range of the quality across the Thousand Genomes Project, there's quite a range, and so it goes from close to 20 all the way up to nearly 50, where I want to say 80% of the data is within between 30 and 40. And so yeah, so this is a value that is computed from a Phred score. It's not directly a Phred score, but it's an average Phred score across the entire genome. I mean, it's not an ideal metric to use. Ideally, we would have a per base, per individual quality value, but those statistics weren't available to us at the time.
Yeah, and the fact that those are not just random errors, but they result in some very specific signatures. That fact suggests that it's not just the low quality bases that are randomly erroneous, but there is something with that sequencing process, or maybe data preparation process, or something, right, in the pipeline probably, that causes this. And by the way, you looked into whether that could be an artifact of sample prep, right? And can you talk about that part of your study?
Yeah, so this was kind of a long-standing unanswered question for quite some time while we were working on this project. Are these actually, false positives that are just turning up in the sequencer, or is this actually, real mutations, but that are somehow affected by, the sample prep? And so just to kind of summarize it, are these spurious mutations, or is it a cell line artifact? And so the cell line artifact hypothesis would be something along the lines of, so you sample your individual, you collect the blood, and then before you can go from blood to sequencing data on your computer, you have to extract the cells, you have to treat them to turn them into cell lines. And so these cell lines are then stored and transported all around the world for research purposes. And a lot of these techniques for sample preparation were being developed at the time as they were doing the Thousand Genomes Project. And so the whole sample prep and sequencing platforms were being updated as they were being sequenced. So that means that some samples would have been prepped with one method, and other samples would have been prepped with another method. And in this cell line artifact hypothesis, what we were seeking to at least rule out was, are these actual physical mutations in cells that could have been impacted by this cell line prep, or is this a technical artifact in the sequencer or in the data processing downstream? And the way that we tested for this is there was 83 individuals from the Chinese Han population that had been very deeply sequenced recently. So the sequencing depth is referring to the number of overlapping reads when you're doing your alignment. And the 1,000 genomes is an average read depth of, I think, 5 or 6. It's pretty low. And then this ultra deep resequencing effort had a read depth, I think, of 60, if I'm not mistaken. So much, much higher. And because we're comparing the same individuals to the same individuals, so we're looking at these 83 individuals that have been resequenced between the 1,000 Genomes Project and this resequenced version of the 1,000 Genomes Project, we were able to compare, are these mutations present in these cell lines, or is it a technical artifact? And so we found that roughly 2% of the variants that we did identify were replicated in the resequenced data, but that meant that 98% of the variants that we had identified were missing from this resequenced data, which is very strong evidence suggesting that these mutations are not actually present in these cells.
Right. So an important thing to maybe clarify here is that what those people doing the 1,000 Genomes Project did was they not just collected some samples, sequenced them, and basically threw them away, but they prepared these cell lines. So they perhaps envisioned that their methods weren't perfect, and they envisioned that in the future there will be better methods. And so once they collected the samples, the biological material, there was some value in preserving that material, and that's basically what you took advantage of.
Yeah, exactly. And yeah, I mean, so there are many reasons to store samples, and one of them you just mentioned is that maybe there's going to be better technology in the future, but there's also, maybe different technology. What if they wanted to sequence the RNA from these cells, or what if they wanted to sequence the proteome data from these cells, or any type of new technology might be of interest to this particular data set. And because of the great value of the diversity and the experimental design of the 1,000 Genomes Project, it was definitely worthwhile to preserve these cells for future work, for sure.
Yeah. On the other hand, so regarding the sample prep hypothesis, the hypothesis that these signatures originated in the cells and then they were genuinely sequenced,
right,
accurately sequenced, right? That hypothesis wouldn't explain the correlation with the sequencing quality, right?
Yeah, so that was something that we were kind of dealing with and trying to make sense of, and no, so the results of our experiment here shows that it is not a cell line artifact, which makes a lot more sense with this data quality metric that we were observing. But it was still something that we needed to rule out.
And so once you established, so let's revisit what you established by this point. So you established that these signatures are fake for the reasons that first it was absent from a different data set and it was absent from the resequenced data set, right? So you were pretty confident that these signatures aren't real, but then you wanted to generalize this onto the bigger data set, onto the whole 1,000 Genomes Project. And so first of all, let's clarify, why is it not simply a matter of running the same regression for the rest of like 25 of the other populations?
Yeah, so that's a great question. And actually, that was what we did initially. So we ran this genome-wide association study, essentially a GWAS, for association to quality in the Japanese population. And we found a certain number of variants that were significantly associated to quality. And then we did the exact same method and applied it to each of the other 25 populations of the 1,000 Genomes Project. And this resulted in finding over 1,000 variants that were significantly associated to quality independently. So that means that each of these variants would have been identified separately in the in each of these populations. But because the Thousand Genomes Project only has roughly 100 samples per population, we were pretty limited with the power of the statistical test we were running. You can imagine that running a genome-wide association study with 100 individuals is not a very powerful study to do, and the fact that we were even able to find any significant associations was kind of surprising. But in order to increase our power to identify more positions in the genome, we had to do a combined test. And the reason why it's more statistically powerful is that if you can imagine a position that is just under the genome-wide significance threshold in one population, and then also just under the genome-wide significance threshold for another population, if you combine these two populations or if you combine this test with these two populations, it's very likely that that single locus that I just mentioned would be very significant because you have twice as many samples, and in both these cohorts, it's slightly associated to quality.
And to come from the opposite side, why couldn't you just combine, all these 1,000 genomes and run a single regression on the whole data set?
Yeah, so the reason why you can't just combine all these samples blindly and run this association test without taking into account population structure is that you might find that, for example, the Japanese population has a large heterogeneity in the data quality of this population, hence why they identified this particular signature in that population. But that means that the average quality of the data from this Japanese population is lower than other populations. So what that means is that you might find mutations that are real mutations that are in higher frequencies in Japanese individuals, but that would happen to be associated to quality simply because they're Japanese. And so this is where the collinearity of the population and the quality comes into play.
Okay, can you explain the term collinearity?
Yeah, so I guess maybe the best way for me to explain this would be it's like a nested batch effect. And so what that means is that when these samples were being sequenced, they weren't being randomly sequenced from different individuals from different populations. There was often an entire population sequenced on one day in one sequencing center. And so what that means is that it's hard to distinguish between mutations that were the result of a technical artifact on that one day versus mutations that were just real mutations but present only in those individuals sequenced that day. And so the collinearity is referring to you have the population structure or the allele frequency of a certain mutation in a population would also be associated to quality simply because all those individuals are sequenced on one day at the same time in the same center.
So you had to develop a statistical model that would strike a balance between these two dangers, not having enough power and coming to the false conclusions because of these spurious correlations, right? So how did you approach building the statistical model?
Yeah, so this is something that is fairly commonly done in genome-wide association studies, where typically people will either use a categorical variable of population, or more commonly they'll use the top 4 or top 5 principal components from a PCA, which would control for population structure in the association test.
And when you say PCA, what do you run your PCA on?
Yeah, so we would typically you run PCA on the actual genetic data that you're doing your association test on. But in our case, because we were trying to distinguish between batch effects and real sequence data and population structure, we had to use the genotype data from the Thousand Genomes Project, which would have been sequenced or which would have been genotyped using a different technology. And so this PCA was computed using those positions to hopefully avoid taking into account these batch effects in the PCA.
Mm-hmm. But like more specifically, so you have this matrix, right? What are the rows? What are the columns?
Yeah, so we have so the rows would be individual positions, and the columns would be an individual from that population. And so typically individuals from the same population will cluster together because they share recent ancestry. That would mean that their allele frequencies are more similar for more positions. And so when you're doing a principal component analysis, you're able to kind of pick up on a lot of this population structure.
And so after you run your PCA, you're trying to find the smaller bases. You're trying to find this, let's say, 5 principal components that would explain the person's genotype.
Yeah, exactly. So I guess the null model for our test is, can we predict an individual's genotype simply based on which population they belong to or they come from, or so that would be for the categorical variable. But in the case of PCA space, for example, we would just say how much of the genotype can we explain through PCA. And so what that means is that we're trying to predict. So this is a logistic regression where we're trying to predict your genotype based on a phenotype. In this case, the phenotype is the data quality. But the null model would just be, how well can we predict your genotype if we know which population you belong to? And then the alternate is, how well can we explain it using population and quality? And then we take the difference in the betas between these two tests, and that is the deviance, which is a chi-square distributed variable that we then use to compute the p-values for each of these association tests.
So in your model, you're not the null model, but the alternative model, you have, let's say, 6 predictors in the logistic regression, 5 of which are the top principal components, and then the 6th is the base quality.
Essentially, yeah, that's how it would work.
Cool. And so, yeah, what did you find once you were able to generalize this to the whole Thousand Genomes Project data set?
Yeah, so once we were able to perform this test on all populations simultaneously while controlling for population structure, we were able to identify a total of 24,390 variants that were statistically significantly associated to quality. I should say that only 15,000 of those actually passed the Thousand Genomes strict mask filter. So that means that this strict mask that I was talking about earlier is actually doing a pretty good job and was able to remove many of these variants simply because of where those variants were in the genome. But that's still 15,000 mutations that are associated to quality that are currently included in the best, up-to-date quality controlled version of the Thousand Genomes Project.
Yeah, that is scary. And so, but like to put this into perspective, what's the total number? Is it like around 1 million SNPs?
There are, give me a second, I have that number. Yeah, so there was a total of 28,516,000 variants included in the test that we ran. And so out of that 28 million, there was 15 or 24,000 of them that were associated to quality.
So that's a small percentage, but still, if those end up, being discovered as statistically significant in some kind of study, and then someone discovers a gene for X, right? That's scary. So, like, who is impacted by this? So obviously, like people who run GWASs, right? People who are looking for these mutational signatures. What are the implications of these fake variants in circulation?
So the first thing that we wanted to look at is maybe the imputation servers are ahead of the game. Maybe they know that some of these variants have issues. And so the first thing that we did was to just test to see if these particular variants are included in imputation services.
And talk a bit about what imputation services are.
So when you so sequencing an individual is still relatively expensive. So that means sequencing every single base of the genome. But oftentimes you don't actually need that super high resolution data, and it's often cheaper to just genotype an individual. And so that's using a different technology. This would be like chip sequencing, for example, where you're only sequencing every 1,000 bases of an individual's genome, or you're sequencing particular SNPs across the genome that you know ahead of time are very good at distinguishing between different people from different populations, or maybe risks for certain diseases. So genotyping data is very common. So one of the steps that people do once they have genotype data is they'll try to get more data out of it. And so this is where imputation comes into play. So if I'm a researcher today who just got some money to genotype individuals, I would get the genotype data, but then try to increase the number of SNPs in my data set by submitting my genotype data to an imputation server. And then this imputation server would then compare the mutations of a given individual to a reference panel. And so, for example, if the sample in question is from the Japanese population, then they might, for a particular region of the genome, they might be very similar to one of the Japanese individuals in the reference database. And so all of the mutations from that individual in the reference database would then be filled in or imputed onto this genotype query, or this sample that you're trying to impute. And so essentially what this means is that these spurious mutations in the 1,000 Genomes Project are being imputed onto new data as it's being imputed.
Mm-hmm. So the idea of the imputation is that there aren't that many haplotypes. There are fewer haplotypes than people, because when someone has a baby, right, they pass their genome mostly unaltered. There may be some recombinations, there may be some mutations, but mostly, at least piecewise, it's the same haplotype as in one of the parents. And so you can sort of create a map of these haplotypes and figure out, right, infer the regions of the genome between two SNPs. By the SNPs, you can figure out which haplotype that comes from, and then sort of fill in the rest as in that sequenced person.
Exactly.
And these imputation servers, who are they run by? Are they run by researchers? Are they run by commercial entities?
The most popular imputation server that is used in research is the Michigan Imputation Server. So this would be run by the University of Michigan, but because they have a lot it's a black box in the sense that we don't know how many samples or which references are used in this database, for good reasons. I mean, this tends to be pretty private data. I mean, sequence data is very private, and so for that reason, we don't know exactly which samples are included as part of the reference panel, and so that was the main reason why we seeked to see if these particular positions were included in the Michigan Imputation Server.
How did you do that? Did you submit the like the normal variants and then just waited to see if the fake variants would be returned by the server?
Yeah, so we did a kind of a worst case scenario approach where we actually imputed the actual 1,000 genomes data,
Uh-huh.
which is kind of a bit of a circular argument because, of course you're going to get really high imputation scores because you're using the actual individuals that are in the reference panel.
Right.
But I mean, that was the point. We just wanted to
Yeah.
see, are these positions going to be imputed onto these samples? And so we found that all of them were. And so when we took the genotype data from the 1,000 Genomes Project and we imputed it using the Michigan imputation server, all of the variants that we identified as being associated to quality were imputed back onto these samples. I should say that it's not likely that all of these variants would be imputed if the individual is not from the 1,000 Genomes Project. So if you were to take any, individual sequence data or genotype data, it's not guaranteed that all of those variants would be imputed onto that individual. It's just because we use the actual reference samples and imputed them onto the genotype data.
Mm-hmm. And in terms of the impact, so let's say those variants are being imputed, and let's say you're running a GWAS study. Do those fake variants have a higher chance of being sort of picked up by a GWAS, or do they just get a fair chance out of those 28 million variants?
Yeah, so we didn't find an enrichment of GWAS hits for these particular variants, and I mean we don't expect any of these variants to be associated to any phenotypes. So we didn't find an enrichment in these variants, but we still searched the GWAS catalog to see if any publications had reported these variants to be associated to some phenotype, just to see whether or not these variants have impacted genomic analyses.
And of course, there have been, right?
Yeah, so we found 17 papers that reported variants that we identified as being associated to quality.
Did you follow up on them? What happened next?
So the next thing that we did was, okay, so first we have to see, well, did they actually use the 1,000 Genomes Project sequence data directly, or were they using it for imputation? And for the most part, they used imputed data from the 1,000 Genomes Project. There was a few studies that used the actual sequence data or some other version of some in-house reference panel imputation. But one thing that was very reassuring, I guess, is that all of these studies used the state-of-the-art, very strict quality filters, including Hardy-Weinberg equilibrium, deviations from an expected allele frequency, or just sequencing quality thresholds. And so, despite using these highest best practices for quality control, some of these variants were still near genome-wide significance for some of these phenotypes that they tested for.
And that's an interesting connection to the Hardy-Weinberg equilibrium. Could you explain what it is and how you could use that to perform quality control?
Yeah, so the Hardy-Weinberg equilibrium is a theoretical expectation from what we know about population genetics. And so essentially it's looking for, so if we know that there is a certain number of heterozygote individuals, we can predict the expected number of homozygote individuals based on the Hardy-Weinberg equilibrium. And so, for example, if we find an abnormally large number of heterozygotes without finding a certain amount of homozygotes, or vice versa, then that particular position would not be within the Hardy-Weinberg equilibrium test.
And so that will indicate that something is shady with that particular SNP.
So not necessarily. So for example, if a position is under active selective pressure, then it would not necessarily respect Hardy-Weinberg equilibrium. And there's also, so for example, sickle cell anemia has a selective advantage for heterozygotes. So you would expect that maybe that the distribution of that particular variant, which protects people from sickle cell anemia, or sorry, that protects people from malaria, would not necessarily be in Hardy-Weinberg equilibrium.
Mm-hmm.
But for most population genetic studies, when you're looking at regions of the genome that are mostly accounted for by drift, you don't necessarily want to include regions of the genome that are under active selection. And so for that reason, people will use Hardy-Weinberg equilibrium tests to remove some of these variants.
Right. So you were talking about those studies that picked up the fake variants and how they were able to filter out some portion of these fake variants using the Hardy-Weinberg equilibrium and other quality control measures, right? But not all.
Mm-hmm.
So some still leaked through.
Yeah, that's right. And so I should also say that for the most part, these studies did not find genome-wide significance for these particular variants, but they were close enough to genome-wide significant that they included them in their supplementary materials.
Mm-hmm.
But this is still important for science in general because many of these supplementary materials are included in polygenic risk score tests where they'll do some meta-analysis combining multiple GWAS studies to see if they can get more power out of these studies by combining them. And so there is a chance that some of these variants, even though they're only somewhat, weakly associated to whatever phenotype is in question, that combining multiple of these association studies in a polygenic risk score might include some of these variants that are spurious.
And then one other thing that you did was to look at how these spurious variants were distributed across the genomes, right? And you found something quite interesting there.
Yeah, so when you look at the distribution of these Q-associated variants across the genome, we found that there was almost twice as many variants in chromosome 1 compared to the rest of the genome. And this is accounting for the size of chromosome 1. So the rate of these variants was higher. And this was a bit of an unusual result. And we kind of had a hard time making sense of it because, why would there be an enrichment in one chromosome? It doesn't really make sense biologically because your DNA repair mechanisms or DNA replication mechanisms, they don't know which chromosome they're working on. So they wouldn't necessarily there's no reason why there would be an enrichment of mutations in one chromosome compared to another. It also doesn't really make sense technologically because when you chop up all the bits of your genome and send it to your sequencer, the sequencer doesn't know which chromosome it's working on. So why would there be an enrichment of these particular mutations in a chromosome? And I guess the only option available left is data processing, which actually kind of narrowed down our search for the cause of these mutations quite a bit. So in a lot of these bioinformatics pipelines for data processing, it's quite common for researchers to split the work on a per chromosome level, where you'll run the same alignment or genotype calling or whatever process, you'll do it on a per chromosome level. And so I should say we didn't actually narrow down exactly the cause of this, the source of these mutations. But our best guess is that it's something in the data processing pipelines of the sequence data.
Right. So you didn't yet get to the root of that, but at least in principle, that's an empirical question and one that shouldn't be too hard to figure out. If you have access to the original sequencing data, right, you can run the pipeline, or you can run your own pipeline or a more modern pipeline, and then you can differentiate between whether those mutations arose in the sequencer, which I think was your main hypothesis because different populations were perhaps sequenced on different instruments, and some older instruments may be producing subpar quality results. And in that case, you would be able to reproduce those signatures using a modern pipeline. Or maybe you wouldn't reproduce those signatures, and then it's a purely sort of bioinformatic pipeline issue.
Yeah, exactly. And so this is almost a kind of a good news in the sense that if it is really an issue with the way that the data was processed, then recalling the 1,000 genomes data would potentially solve those issues.
And then you have another piece of good news, right? I believe the New York Genome Center is doing something exciting in this space.
Yeah, so in response to the preprint that we had released for this paper, the New York Genome Center, so the news spread and people were kind of talking about, it's weird that nobody has resequenced this data set because it's so valuable to the scientific community. And then, somebody else said, oh, I thought the New York Genome Center was doing something like this. And then, later on, the New York Genome Center made an announcement saying that they were, in fact, in the process of resequencing the entire 1,000 genomes data, and from scratch using brand new technologies. And so this is still unpublished data, but the data is publicly accessible. And so I would expect that maybe a version 2.0 of this database would be updated with their publication that is forthcoming. But this is, really great news, and it just goes to show how quickly science can move forward when you just put out a preprint and, your peers are the ones that are actually moving science forward. It was a really kind of a happy ending to this story where for the longest time we were thinking, should we recall the sequence data? Should we, do all this extra work? And then it just so happens that another research group had already found the solution to all of our problems, despite not necessarily knowing the issues with the older versions of the 1,000 Genomes Project.
Yeah, and on the other hand, even once we have the more modern data, both in terms of the sequencing instruments and in terms of the pipelines, I think we shouldn't get too complacent, and we should remember that, people back then also perhaps didn't anticipate that there would be such severe issues with their data. And so your work could become another quality assurance step, quality control step in the future, even if we have better instruments, better pipelines. Still, this is an easy step that we could run this kind of logistic regression to see if we get some unexpected correlations with the base pair quality.
Exactly. And so I should also mention that there have been a few other groups that have identified issues with the 1,000 Genomes Project. Most notably, Mafisoni et al. From 2018 reported. So they actually, and this was actually really interesting for us to read, because when we were reading the paper, the headline was, patch effects in the 1,000 Genomes Project. And we thought that we had been scooped because, this is exactly what we were working on. But they actually approached this in a very different way, which was actually really interesting to read, because I knew so much about this database, and then here's this completely different approach to finding, a similar issue, where they looked at linkage disequilibrium associated variants. So, for example, two variants on different chromosomes are not very likely to be linked because they're on different chromosomes. So there's basically no linkage disequilibrium for these. But they were looking for unusual linkage disequilibrium patterns in the 1,000 Genomes Project. And they found, several thousand of variants that were unusual in their linkage disequilibrium patterns. So yes, I would definitely suggest to run our test of this simple logistic regression on any database because it's a really easy test to run. You're basically doing a GWAS on quality. But I would also highly recommend using the method that Mathesoni et al. Used because they identified a different set of variants. There was some overlap, but they were better at identifying some variants, and we were better at identifying other variants. But clearly, the combination of these two methods will do a great job at removing a lot of these spurious variants.
Very cool, Luke. Great job. And thanks for your time. Thanks for coming on the podcast.
Yeah, thank you for inviting me. It's really great to hear that people are interested in this research. And I definitely find myself kind of getting in my own bubble and not thinking that anybody's actually interested in this work. And it's just really great to hear people are interested in this. So thank you for inviting me.